That's it. It's 5 o'clock in Copenhagen. What time in Denver?
It is 9 a.m. in Denver.
Nice and early. Welcome to work.
Thank
So,
you.
anyway, welcome everybody to the 8th webinar of this webinar series that started due to, strange times in the world. So, today we have Fabian with us on the 102, as we said 101 was last time. And he's had a few weeks to prepare for this webinar now. And again, I'm Anne-Catherine, one of the co-founders of Nordic Laboratories and DNA Life. And we try and help practitioners to make it easy for them to get hold of lab tests and supplements around the world. And especially these days where we see a lot of patients online, it's easy because we drop ship both test kits and supplements to the patients. And then before we dig further into Tom's talk, I want to announce the next webinar next Thursday. I had to quickly rearrange what has to happen next Thursday. So I had one plan and now I have another plan, and it was just arranged like 2 minutes before we went on air here. But next time it's going to be with Dr. Wendy Musasari. I might pronounce it wrongly, but we will get the lecture next time and how it's pronounced. She's a PhD and her specialty is in antibiotic resistance. And she's going to go into much more like environmentally issues in regards to antibiotic resistance. And that of course leans up a little bit to what we are talking about today, but we are going to dig even deeper then. So, and of course, today is being recorded and the recordings will be shared as soon as possible, as soon as the internet and Zoom allows for that to happen. But enough from me. Tom, who's also a doctor, PhD in gut and microbiome and so on, and nutrition as well, immune system and brain health and so on is the specialty. So it's a real pleasure to have you live again here. I've been looking forward to it since last time because you are a really, really good speaker, and I really, really enjoyed it last time, and the feedback was really, really good. So Tom, I'll pass on the stage to you. Enjoy.
All right. Well, thank you so much, Anne-Catherine. I really appreciate the opportunity to be here today, and I hope everyone is having a great day and staying healthy and staying safe. So the topic today is kind of a follow-up to what we talked about last time. So last time was more of an introduction to the GI map test, sort of an overview. This time we're going to focus on kind of extending that information that we went through the first time and focusing more on the pathogens and opportunists on GI map, in particular focusing in on a few specific examples. And what we really need to understand about these pathogens is not so much just the pathogens themselves and what they can do in terms of potential impacts on health, but also how they interact with the ecosystem. And that's really important for getting the most out of interpreting GI map and using that information to help patients. So again, that's going to be the focus today and building off of what we went on last time. All right, so in this next slide, this is one of the slides that we had on the last webinar, and it's just kind of an overall representation in terms of the markers that were carefully selected for GI map. So we have markers that reflect both the microbial ecosystem, as well as markers that reflect what's going on with certain key aspects of GI physiology. And that's especially important for understanding what's going on with patients and connecting the dots. So again, traditionally the focus has mostly been on individual pathogens, sort of generally recognizing dysbiosis, those sorts of things. This information is really taking that much further and really combining that with the latest research to help you get the most out of GI map and understanding what's going on with your patients. So this is kind of a key concept that I wanted to really focus on today in terms of kind of a key theme. So this idea that we have on the test, some of the key pathogens that can affect the gut, as well as various opportunists. And then we also have some of the normal microbes in the microbiome as well. So that combination of normal microbes that may be out of balance, plus the opportunists, and also whether there are pathogens present, really is what constitutes dysbiosis. And we kind of hear that term in a very general sense, that you either have dysbiosis or you don't. And there are some general sort of common characteristics of dysbiosis, but there really are many different types of dysbiosis depending on the specifics of what's going on with an individual patient or client. So that's really important for taking a personalized approach and really understanding what's going on with a specific individual. And then that really interacts quite a bit with the gastrointestinal physiology. So as you can imagine, we tend to think of pathogens coming in and causing problems and then affecting the health of the gastrointestinal system, and then possibly systemically as well. But it actually goes in both directions. We're learning more and more about the conditions in the gut, and especially when these conditions are imbalanced, that can promote the growth of pathogens and opportunists. So we're really learning quite a bit about this two-way interaction. We're going to go through some key case examples that illustrate this interaction. And then that's really where you can get the most information, because it's not only understanding the impact of the pathogens, but if you're looking at the physiology, that can help you understand why the pathogens potentially are there in the first place and what they might be doing. So this is just kind of a reminder, a visual that we're talking about a lot of information and concepts, but in reality, it's definitely helpful to kind of picture and keep this picture in your mind of the gastrointestinal tract and what you've learned about physiology and anatomy, because what we're seeing in a typical stool test like GI map is really, in a sense, kind of the output from the GI tract overall, primarily from the colon, but we also do pick up organisms because this is a DNA test. We can pick up organisms that are coming from higher up in the GI tract. Kind of a prime example that we talked about last time would be H. pylori in the stomach. And again, because this is a DNA test, we can pick up that organism, whether they're dormant cells, store cells that may still be active, or even DNA from those organisms. We can pick that up in stool. Now, just kind of a side note, that's a common question that we get about, does DNA detect past infections? And the general answer is no. DNA is generally not going to be hanging around in the GI tract for long, mostly because just the material is passing through and usually passes out within a couple of days. So we're generally not detecting past infections with DNA, we're detecting what's currently there. So moving on to the next slide. So this is basically the three common dysbiosis patterns that we tend to see. So this is kind of a high level look at the various patterns that we see, and then connecting that to how that relates to physiology. There certainly are variations and specific details within these patterns, and potentially other patterns as well, but these are the three most common patterns. We went through some examples last time, especially for that last pattern, the digestive dysfunction dysbiosis pattern, which is the most common pattern that we tend to encounter. And again, if you're thinking about the GI tract and kind of thinking of the flow from upper GI tract down to the lower GI tract, there's sort of this domino effect. So it's important to consider the impact of digestive dysfunction on what's going on in the rest of the GI tract, especially downstream. We talked last week about H. pylori and some of the impacts of H. pylori. So I just want to kind of recap that here real quick, because H. pylori is definitely one of our most clinically relevant organisms. So we tend to see H. pylori elevated. It's often just present. And when we pick it up on the test, it's usually already imbalanced. But especially when it's high, when it's flagged as high, it can be especially impactful on the GI environment, and in particular on affecting stomach acid. So this is a review article that I highlighted in the last presentation. And this is just kind of a reminder quote from this in terms of the key impact of H. pylori. And it's really important to understand this because that can set in motion a lot of imbalances within the GI tract. So this quote is, most patients chronically infected with H. pylori manifest a pangastritis with hypochlorhydria. So that last part, the hypochlorhydria, really has potentially a lot of impacts based on what we see clinically, and certainly what we know from research as well. So again, this really kind of leads to a domino effect where low stomach acid can predispose to pathogens. We'll see some cases where there's evidence for that. It can certainly set in motion poor digestion downstream, as well as dysbiosis, and then those can have further impacts. So again, just kind of a reminder, of course, H. pylori is mostly present in the stomach, and then there is evidence that it's also potentially present in some individuals in the duodenum. And again, that can have a big effect on the pH downstream, particularly in the small intestine, especially the upper small intestine, where the dysbiosis that can lead to in the small intestine causes further issues. We'll actually look at a specific example of that as we go through the webinar. The other thing that we emphasized last time is that on the test, we can often see a potential impact of H. pylori in terms of an overgrowth pattern, which we tend to see both in the normal bacterial flora section. It tends to vary from person to person somewhat, but oftentimes we'll see high Clostridia, high Firmicutes, et cetera. This is just kind of a reminder because we are going to see some of these patterns repeated in some of these cases that we're covering today. Opportunistic bacteria is also a section that we're going to see a lot of these imbalances, generally an overgrowth pattern that tends to result from altered digestion, low stomach acid, and oftentimes caused by H. pylori. So again, the consequences tend to be changes in pH, food availability that tend to affect this microbial balance and leads to the overgrowth. It also leads to increased microbial fermentation. So as you can imagine, when foods are not digested, typically we think of carbohydrates not being well digested that leads to increased fermentation by the bacteria in the gut. And then that can lead to the production of excess gases and microbial products. And those can have a big impact on GI physiology as well as symptoms. They can also cause localized inflammation, especially in the small intestine, and in some cases in the large intestine as well. And that also can lead to leaky gut. So there's a lot of evidence linking inflammation to leaky gut. And of course, that all can lead to various symptoms and possibly diseases in some cases. So again, this brings us back to this overall picture here where we went through this last week and talked about H. pylori and low stomach acid. So that's kind of an example of sort of an opportunist / pathogen. There are pathogenic strains that express virulence factors that can have more significant consequences. But even just the typical H. pylori that's non-virulent, that doesn't have the virulence factors, can still have this impact on stomach acid, which then impacts gastrointestinal physiology, impacts the ecosystem that can then further impact the system. So I want to go through a quick example here, and this is related to H. pylori, but starting to focus more on some of these other pathogens and opportunists that can be part of that picture in many patients. So this case is focused primarily on C. difficile. So this case involves a 49-year-old male. Just kind of a very quick synopsis of the key health issues. The prominent symptoms were consistent nausea, abdominal pain, and then frequent loose stools. And when we look at his GI map results under the pathogen section, we can see that the two markers for C. difficile, so in this case, we're looking at the genes that encode toxin A and toxin B. So that's confirming that the pathogenic strain or strains of C. diff are present in this individual, and that these strains do contain these key toxins that tend to produce the main effects of C. diff. Now, something that's important to note, since this is a DNA test, we're not testing directly for the toxins. So we do know that pathogens can be present. Pathogenic strains can be there that have the genes to encode toxins, but that doesn't necessarily mean they're expressed. So that's something that's important to know, because sometimes you may see that these pathogens are elevated, and in some patients, we don't see that they have any significant symptoms in terms of the typical symptoms associated with that pathogen. So there's a lot of factors that are involved in whether or not these toxins are actually expressed. But it is confirming that they potentially can be expressed and that the pathogenic strains are present. And especially the thing to note here is when they're flagged as high, that means they're growing pretty vigorously in the gut, which basically indicates that the conditions are favorable to their growth. And we're actually going to go through some of those specifics here in this webinar. Also on the report, we can see that H. pylori is positive here. It is detected. It wasn't flagged as high, although in this case, it's right at the cutoff. Just a side note is that we often do see H. pylori being clinically significant, even if it's not flagged as high. So generally, the higher the number, the more the overgrowth is at the time of sampling, and that can be pretty significant. So again, even if it's not flagged as high, it's always good to check the H. pylori just to see if it's even present, because that can still have an impact on stomach acid. In this case, only one virulence factor was present, indicating that the strain or strains that are present are not necessarily extensively pathogenic. Typically, that situation is when the virulence factors are largely positive for most, if not all of them. So H. pylori is present, probably suppressing stomach acid. We also see kind of this typical overgrowth pattern in the normal bacterial flora section, including Clostridia, which is the E8 level, and then Firmicutes, which is at the E11 level. Both of those are flagged as high. Those are two markers that are commonly elevated with H. pylori. We can also see there are a couple that are low. So Bacteroidetes under the phyla is flagged as low, and then Akkermansia is low as well. So we'll come back and talk about how these may relate to C. diff. So that's one of the key things to learn about interpreting stool tests in general, and especially GI map, is not necessarily looking at all these markers individually and looking at just C. diff and noting that, and then noting that they have dysbiosis. We want to make those connections based on what we know clinically and from research to help us kind of flesh out that picture of what's going on in the gut. So then under opportunistic bacteria, we can see a pretty significant overgrowth pattern. So this supports an impact of H. pylori. It's very consistent with what we know about the impacts of hypochlorhydria. So definitely the picture looks like there's likely to be hypochlorhydria. Under autoimmune triggers, the two that are detected here are perfectly normal in those ranges. So those are not notable. We do see that Candida is also present at high levels. That oftentimes is part of this digestive dysfunction related pattern, and it may be caused in part by H. pylori and this sort of downstream dysbiosis pattern, plus the lack of digestion and absorption. That may be a scenario that helps to fuel the growth of Candida. We don't see any parasites present. Under the intestinal health section, we can see that under digestion, that aspect of digestion looks fine. So steatocrit, again, is fat malabsorption. Elastase is an indicator of pancreatic function. We're looking for levels ideally around 500 and above, so this looks pretty good. The GI markers look good, nothing out of range here. The immune response, the secretory IgA, looks to be well within normal ranges. Antigliadin is just a little bit higher than we'd like to see it. Anytime we see it around 100 and above, that's mostly something we see with this overall digestive dysfunction pattern. Again, might indicate a tendency towards gluten sensitivity. But clearly the key thing on this section that's notable is the high calprotectin. So that's indicating that there's a high level of inflammation present at the time of sampling in the colon in particular. And just another side note on this is that calprotectin is best at reflecting inflammation in the colon, less so in the upper GI. So you can, for example, you can have a pretty significant H. pylori infection in the stomach that's causing inflammation in the stomach, but we don't necessarily see calprotectin being elevated. So again, it's mostly a marker reflecting inflammation in the lower GI tract, but that's clearly the case here. So what do we make of all these markers? And we see that C. diff was present. The question here is it having an impact in the gut based on what we can see here? And what may be predisposing to the C. diff overgrowing? So again, this is just kind of recapping the case. Again, this is all from a male that's 49 years old that complains of nausea, abdominal pain, and loose stools. The case summary is that we do see that C. diff is elevated along with high calprotectin. So that's one of the ways that we often gauge whether C. diff seems to be having an impact. We know C. diff infection can cause inflammation. If we don't see inflammation, which is a scenario that we often see with C. diff as well, that may suggest that it's having less of an impact. But in this case, certainly it's a strong suspect for causing that elevated inflammation. We also see that H. pylori is present, Candida, and the overall digestive dysfunction pattern as well. And then we saw low Bacteroidetes and Akkermansia, so low beneficial bacteria. So let's talk a little bit about how these may be connected based on what we know from research, and also what we have seen quite a bit clinically as well. So the title here of this article is Proton Pump Inhibitor Therapy and Risk of Clostridium difficile Infection. systematic review and meta-analysis. So they say this meta-analysis provides further evidence that PPI use is associated with an increased risk for development of CDI infection or Clostridium difficile infection. There's a lot of literature on this that suppressing stomach acid in various ways can increase the risk for various pathogens. So in one sense, that makes perfect sense, intuitive sense, because that is one of the key protective barriers if we're being exposed to pathogens through foods, through environmental factors. When we swallow those, they go through the stomach. Usually they're pretty efficiently killed off in most cases by stomach acid. For C. diff, it's a little bit of a different scenario because typically it produces spores. And so the main way that we're exposed to C. diff is through spores, which are much more resistant to things like stomach acid. So technically they can pass through and still be viable by and large. But studies have shown, which I'm not going to get into here, it's kind of beyond the scope of this webinar, but studies have shown that basically low stomach acid increases the germination of those spores. So that can lead to a higher level of the C. diff growing in the gut. So kind of a quick summary that it has been shown through multiple studies, multiple lines of evidence, that low stomach acid can predispose to C. diff infection. So we certainly see a potential connection there with H. pylori. So let's talk a bit about the supportive approach. So it has been shown that antimicrobial botanicals can help to reduce C. diff. Certainly antimicrobial botanical protocols have been developed for H. pylori and Candida. So those may be quite helpful in overall helping to improve the dysbiosis picture and helping to counteract C. diff as well. Lots of good evidence, both from research and clinically, that Saccharomyces boulardii can also counteract C. diff, H. pylori, and Candida. So that's often something that's included in protocols in this type of scenario. And then this is something I'm actually going to talk about some of the research behind it, but now there's a little bit more focus on dietary modifications. We know, for example, that high fiber diets can potentially reduce C. diff as well. There's new evidence that lower protein diets, at least temporarily during treatment, may also help to reduce C. diff growth and infection. And in general, whenever there's inflammation present, certainly anti-inflammatory approaches may be helpful as well. So these are some of the studies kind of backing up these types of approaches to supporting a better microbiome profile when C. diff and H. pylori are present. So the title here is Microbiota Accessible Carbohydrates Suppress Clostridium difficile infection in a Mouse Model. So just an FYI is that their term for fiber, which is what we typically refer to it as, is technically microbiota accessible carbohydrates. So that just translate that into, that's equivalent to fiber. So they say using a model of antibiotic-induced Clostridium difficile infection that typically involves or resolves within 12 days of infection, we demonstrate that fiber-deficient diets perpetuate Clostridium difficile infection. We also show that C. difficile burdens are suppressed through the addition of either a diet containing a complex mixture of fibers or a simplified diet containing inulin as the sole fiber source. So there's a lot of additional evidence from other studies as well. This particular study was a very well done study by some prominent researchers in the field of microbiome science in a pretty prominent journal as well, Nature Microbiology. So this suppression of C. diff infection with fiber is certainly something that's becoming more and more established. We also know that potentially excess protein may stimulate the growth of C. diff as well. So the title here is Clostridioides, which is actually a newer term for Clostridium difficile. So I don't want to get too confusing here, but you may see this term. They've actually reclassified Clostridium difficile. It's now officially referred to as Clostridioides difficile. So just keep that in mind if you see that. That means the same thing as C. diff or Clostridium difficile. So again, the title is C. diff uses amino acids associated with gut microbial dysbiosis in a subset of patients with diarrhea. And they say in the article, the dysbiotic gut microbiota of patients with diarrhea was characterized by an increase in free amino acids, especially proline, and showed increased susceptibility to C. difficile infection after transplant into germ-free mice. So again, they're taking the microbiota from C. diff positive patients that are symptomatic, that have diarrhea, and they showed that they have an increase in these amino acids, and a particular amino acid, proline, in the stool, and that basically conferred an increased risk or susceptibility to C. diff infection after transferring that to mouse model. They go on to say that dietary proline is not essential for humans, and protein-based dietary interventions are partially protective in animal models of C. difficile infection, suggesting that a proline-deficient diet could be a simple, actionable therapy for providing protection to at-risk individuals. So again, this is really emphasizing this kind of new recognition that diet and sort of these physiological conditions in the gut really do have a big impact on the growth and possibly the virulence of some of these pathogens. And this suggests that possibly restricting proteins overall, and especially foods that are high in proline, may have a positive effect on C. diff. Now, the flip side of this is it's not always just dietary content, of course, but also the efficiency of digestion. So again, along with low stomach acid, one of the main impacts tends to be on impacting protein digestion in particular. And that's partly because stomach acid, so acid in the stomach, is required for conversion of pepsinogen to pepsin. And pepsin plays a pretty important role in protein digestion. So with low stomach acid, then protein digestion in particular is often compromised. And then those undigested proteins and amino acids can get into the colon. And then that's a prime environment for promoting the growth of these types of pathogens. All right, so they go on to say here, this is kind of following up on what I just said. So they say recent studies have shown that fecal amino acids positively correlated with the severity of Crohn's disease as well. This indicates that increased amino acid availability may be an important mechanistic effector in certain gut microbial dysbiosis-related pathologies. So long story short, there's a growing list of studies that are showing that increased protein presence in the colon seems to correlate in general with dysbiosis, and especially inflammatory type dysbiosis, and has been shown to play a role, or at least that there's an association in a variety of different inflammatory diseases such as Crohn's disease. So that's a great example of this interaction between the pathogens, opportunists, et cetera, that are present in a particular individual, how they impact physiology, and then how that altered physiology can go back and further impact dysbiosis and other pathogens, et cetera. And so that can sometimes, in certain patients, become a vicious cycle. Okay, so moving on to the next example here, I want to talk a little bit about Pseudomonas, which is one of the markers in our opportunistic bacteria section. So this is a case study that highlights a patient that had overgrowth of Pseudomonas, along with some other interesting findings. So we're going to talk about how this is also linked into these particular symptoms. So this is a 62-year-old female. Basically, it's been confirmed through food sensitivity testing, elimination diet approaches, et cetera, that she has extensive food sensitivities, also complains of sort of unexplained weight gain. The clinician that has worked with her suspected SIBO initially, although the breath test was negative. So SIBO-like symptoms, but no evidence of SIBO. They did go ahead and try a low FODMAP diet, but unfortunately there was no significant improvement. So they were kind of stumped at this point in terms of what to do. And this is really where GI map tends to shine, because we can really get some insights into some of these specific imbalances that may still be a problem for people that have SIBO-like symptoms, but basically were not either confirmed to have SIBO or were not susceptible to treatment. So the results for this patient, we're looking at the pathogens section on the first page. Nothing was detected here, so that looks good. We also see that the parasitic pathogens, viral pathogens, et cetera, were negative, and H. pylori in this case was also negative. So far, everything looks good. Normal bacteria flora section. We do see a few imbalances. So on the surface, this may not look that significant, but we do want to kind of scrutinize the individual results here. And we'll mostly come back to this, but just want to highlight here that one of the markers, Enterobacter, is elevated. That's one of the groups that we tend to see elevated when digestion is not optimal, but it also can be something that is elevated in inflammatory types of dysbiosis. So that's just something to note. Akkermansia, as we talked about in the last webinar, is a really important species in the microbiome. Depends heavily on adequate mucus in the colon for its main food source. So when it's deficient, it can be a sign that that mucus layer is deficient and that the overall intestinal lining health in the colon may not be as healthy as it should be. Also, one of the general conditions that tends to lead to lower Akkermansia can be inflammation. We can also see here that Bacteroidetes at the phylum level is low. That's generally, of course, a very important group of largely beneficial bacteria that also produce various factors that can be healthy for the colon. So that's what we're seeing mostly in the normal bacterial flora section. We do see that there's an overgrowth of opportunistic bacteria. So this is fairly similar to what we typically see with an H. pylori pattern, although H. pylori was negative in this case. But this may still suggest that this patient might have some hypochlorhydria, even if it's caused by something else, in this case, not H. pylori. A couple things to note here is bacillus being elevated is fairly typical. Streptococcus elevation is fairly typical with digestive dysfunction, dysbiosis. But we do see that in this case that Morganella and especially Pseudomonas are elevated. So we're going to focus again mostly on Pseudomonas here, but these two groups tend to be also more inflammatory. And especially Pseudomonas, there's a lot of research around it that's been coming out, indicating that it can predispose to certain issues. So we'll talk about those momentarily. Under autoimmune triggers, again, nothing stands out. The two that are detected are perfectly normal. No fungi yeast, no viruses, so that looks good. Also no parasites. So mostly what we see is this sort of overgrowth pattern under the opportunistic bacteria and also some imbalances in the normal bacteria. Looking at the intestinal health markers, so on first pass, nothing is flagged as high or low. So overall on the surface, that looks good. But if we scrutinize these numbers a bit, looking under the digestion section, steatocrit looks good. It's below detection limits and no evidence of fat malabsorption. For elastase, we do see it's trending low. So optimal for elastase is around 500 and above, and our cutoff for officially being low is 200. But we do tend to see that when there's a trend towards the low end of the normal range, that's still often clinically significant. So generally when it's below about 300 or so, that gets into a range where we do see that that can have an impact and generally supports the digestive dysfunction type scenario. GA markers look good, nothing out of range here. Immune response markers, we do see that the secretory IgA at 534, again, is very close to the cutoff for being considered low, so that would be low normal. That's something we just typically see along with low beneficial bacteria. So that's typically one of the key causes of low secretory IgA is low beneficial bacteria. So that indicates potentially where some of the emphasis should be in a therapeutic protocol is building up those beneficial bacteria to help boost secretory IgA. We also know that deficiency in secretory IgA also can increase susceptibility to pathogens, increase susceptibility to dysbiosis, and also is a scenario that leads to an increased risk for food sensitivities and things like that. Okay, so this is just kind of a recap of this particular patient and the symptoms again. So again, this is a 62-year-old female, has extensive food sensitivities and complains of unexplained chronic weight gain and SIBO-like symptoms, but tested negative for SIBO. And also a low FODMAP diet did not help. So just kind of a side note is we do see a lot of these types of cases where there are SIBO-like symptoms and either they confirmed SIBO and then there was SIBO treatment that was not very effective, or they were suspected of having SIBO, but then the test was negative. So that's sort of a confounding scenario because it seems like they have SIBO, but it doesn't really pan out as far as that is necessarily a correct diagnosis in that case. We do often see that Pseudomonas overgrowth is one of the issues that's common in many of these types of patients. So let's kind of just summarize what's going on with this patient first, and then I'll talk a little bit about the details around Pseudomonas. So overall, we see that this patient has opportunistic bacterial overgrowth, especially of Pseudomonas, relatively low elastase, and those two are probably connected. I'll talk about that a little bit more in a moment. And then also relatively low secretory IgA, which we noted. So this study is really highlighting some of the new research that's coming out about Pseudomonas, linking it to issues with digestion and increased risk for food sensitivities. So the title here, duodenal bacterial proteolytic activity determines sensitivity to dietary antigen through something called protease activated receptor 2. So some of these pathogens and opportunists like Pseudomonas produce proteases. Some of those can have negative effects. We'll talk about that here. So it says, we have recently shown that undigested gluten can be metabolized by certain opportunistic pathogens, such as Pseudomonas aeruginosa, leading to the production of peptides that more easily translocate the mucosal barrier with retained immunogenicity. So kind of translating this, essentially what they're talking about here is that Pseudomonas breaks down through this protease that it produces. It breaks down gluten and also presumably other food antigens. And it can break them down in a way where they're more easily crossing the intestinal barrier, but they're still able to stimulate an immune response. Under ideal circumstances with optimal digestion, those proteins are fully broken down into amino acids so that they don't have the ability to really stimulate the immune system in that way. So really anything that interferes with digestion, whether it's hyperchlorhydria, whether it's these types of opportunists, they're breaking things down in a way that makes them more immune reactive. All of that can increase the risk for food sensitivities. They go on to say results of the study demonstrate that proteases expressed by opportunistic pathogens impact host immune responses that are relevant to the development of food sensitivities independently of the trigger antigen. So they're just referring to here that there are previous studies linking Pseudomonas to gluten sensitivity and also potentially to exacerbating celiac symptoms as well. But they appear to actually play a role more largely or more broadly in food sensitivities. So that kind of relates then to the low elastase. So this is another one of those sort of vicious cycles where if you're not digesting well, whether it's evidenced through low stomach acid or low elastase, then those undigested proteins actually can promote the growth of these pathogens. And then they in turn, by producing these factors like proteases, can make that situation worse and then promote food sensitivities. So it has been shown, for example, for Pseudomonas, mostly in lab experiments where they're growing it in the lab, that it tends to grow very well on proteins in general, but also particularly on gluten and also proteins from dairy and possibly eggs. So that, of course, suggests that when you're trying to address Pseudomonas, it may be helpful to reduce those sources of proteins. They go on to say, thus, correction of bacterial proteolytic imbalance in the small intestine may constitute a new therapeutic target to prevent or ameliorate food sensitivities triggered by specific protein antigens. So this is a brand new area of research that's really been coming out in the last few years, indicating that dysbiosis in the upper GI tract is particularly important for predisposing to things like food sensitivities. And fortunately with the DNA test, we are able to detect some of these types of organisms that are coming from high up in the GI tract. So let's talk a little bit more about Pseudomonas and the connections to sort of common symptoms, common conditions. So we know from research and also clinically that it's commonly linked to food sensitivities, and we do see this a lot clinically. It leads to SIBO-like symptoms that may actually not be SIBO itself. So that's one of the confounders with SIBO, and as we're learning more and more, because we know a lot of people don't respond to SIBO treatments, et cetera, we're finding out that it's because of these other organisms that may be playing a role that aren't necessarily always well addressed by SIBO treatments. Pseudomonas overgrowth can also lead to reduced digestion and absorption. That, of course, can impact nutrient availability as well. It can lead to possible upper GI inflammation as well as leaky gut. So one just kind of quick side note is this patient also complained of excessive weight gain that seemed to be unexplained based on their diet. And so this chronic inflammation sort of scenario, leaky gut scenario, is something that's also been linked to obesity. So that may be playing a role in this individual's inability to lose weight. So in terms of addressing Pseudomonas, and I'll get into some of the details here backing this up, certainly we know that antimicrobial botanicals can be helpful, especially the broad spectrum types of botanicals. Considering avoiding gluten, dairy, and possibly eggs, at least during the treatment period, if not longer term, to help prevent its regrowth. But we do know that there's good experimental evidence that gluten and dairy proteins in particular can promote the growth of Pseudomonas. There's evidence that being insufficient for vitamin D may play a role as well in promoting pathogens and opportunists like Pseudomonas. And then, of course, probiotics. So those can have activity in counteracting many of these sort of negative activities from pathogens and opportunists. So I'll go through some of the research here backing this up. So this study here is titled Effects of High Doses of Vitamin D3 on Mucosa-Associated Gut Microbiome Vary Between Regions of the Human GI Tract. So in this article, they say vitamin D3 supplementation changed the gut microbiome in the upper GI tract, particularly in the stomach, as well as in the duodenum. We found a decreased relative abundance of gamma proteobacteria. That's sort of a group of bacteria that tend to be more inflammatory. They said we found a decreased relative abundance of gamma proteobacteria, including Pseudomonas species, as well as Escherichia shigella species, increased bacterial richness. Basically, vitamin D supplementation may have a positive effect on reducing these types of opportunists. The next article I wanted to highlight here is titled Duodenal Bacteria from Patients with Celiac Disease and Healthy Subjects Distinctly Affect Breakdown or Gluten Breakdown and Immunogenicity. So they say lactobacillus species from the duodenum of non-celiac disease controls degraded gluten peptides produced by human and Pseudomonas aeruginosa proteases, reducing their immunogenicity. So what they're talking about here is we already knew from these other studies, and in this study as well, that Pseudomonas tends to worsen the situation with gluten. So it tends to make gluten sensitivity worse, celiac disease worse, because of the way that it breaks down gluten. Lactobacillus, on the other hand, also can break down gluten, but it does it in a way that actually makes them less likely to stimulate the immune system. So this is really coming back to that microbial balance, in this case referring to the balance in the upper GI tract, where we'd like to see a greater balance of lactobacillus and reduced Pseudomonas, and that may help improve symptoms. Just another example of probiotic type bacteria that can help also address this scenario. So I'm not going to get into the details here, I'll just read the title, which is a commensal bifidobacterium longum strain prevents gluten-related immunopathology in mice through expression of a protease inhibitor. So these different bacteria, probiotic bacteria, and also just the normal healthy microbes in the gut, some of them may produce factors like this that actually can inhibit some of these negative factors produced by pathogens and opportunists. So again, it comes back to that balance and trying to promote the balance towards beneficial bacteria and away from the dysbiotic bacteria. So this again is another great example of this interaction between pathogens, opportunists, dysbiosis, and then the altered gastrointestinal physiology. So in this case, we saw evidence that potentially with lower stomach acid and reduced digestion, mostly in evidence from low elastase, that can promote the growth of Pseudomonas. And then in turn, once Pseudomonas overgrows, mostly in the upper small intestine, it can have a negative impact in causing inflammation, causing leaky gut, setting the stage for increased food sensitivities and other issues. Okay, so again, this kind of brings us back to really recognizing these common dysbiosis patterns. We know that lack of beneficial bacteria can have an impact. We just talked about a specific example there. Inflammatory dysbiosis, so again, Pseudomonas can cause inflammation in the upper small intestine. And also this general digestive dysfunction dysbiosis, which tends to just fuel the whole set of imbalances. So these three different dysbiosis patterns often are interrelated. We don't always see all three of them in all patients. Sometimes we do, sometimes we just see one of the patterns. So this is the next case example. I think I didn't change the title here, so this is not actually focused on Pseudomonas. This is more inflammatory dysbiosis. So this is a case that involved a 22-year-old male who's generally fit and health conscious, but developed diarrhea and abdominal pain at a certain point and has experienced that off and on, especially since he had traveled internationally. So that's always kind of a red flag that possibly they acquired some sort of pathogen when they were traveling. So this is an example of what I was just talking about, that some patients may have all three of these types of dysbiosis patterns present. So we look at his first page of the GI map, so the bacterial pathogens, we actually see that there are two bacterial pathogens that are elevated and flagged as high. Now that's the enterohemorrhagic E. coli and then the Salmonella. There are patients that can have these pathogens, so this looks like a pretty bad scenario with two pretty significant pathogens at a high level. He wasn't really that symptomatic. He just had sort of chronic diarrhea and subabdominal pain. So every patient can be a little bit different in terms of how they respond to these levels. But certainly two pathogens were confirmed. We also see that H. pylori is present at kind of a moderate level. So it's not flagged as high, but is at the E2 level. That can still be clinically significant. And again, whenever we see pathogens present, it's certainly a possibility that stomach acid is too low because of H. pylori, allowing these pathogens more easy access to colonizing. Be some issues related to possibly H. pylori. There may be an impact from these pathogens as well. We also can see that there are two beneficial bacteria or groups that are low. So at the phylum level, we can see that the Bacteroidetes is quite low. And there actually are newer studies that came out in the last year or so showing that specific factors that are produced by Bacteroidetes help protect against certain pathogens like Salmonella. So we certainly know that that can also be another factor leading to the overgrowth of these pathogens, is the lack of these beneficial bacteria in their products. Just above that, we see that the Faecalibacterium prausnitzii is less than detection limit. So that's a very important species in the microbiome we talked about in the last webinar that produces butyrate. Butyrate is a really important short-chain fatty acid that's also been shown in many studies to help counteract pathogens. So there are lots of good studies, for example, especially with Salmonella, showing that butyrate helps to counteract Salmonella. So once again, we have all this evidence that H. pylori is present, that can predispose to pathogens. We have a lack of certain key beneficial bacteria that can also predispose to pathogens. Under opportunistic bacteria, we do see that there's some overgrowth, so that's sort of further supporting this scenario of not digesting well and also possibly low stomach acid. Looking at the intestinal health section, we do see that there are some significant imbalances in this patient. So he does have two significant pathogens. We do see that there is some occult blood that's detected. The FIT level is at 18, so that's officially high. We also see that calprotectin is elevated, so there is confirmed inflammation in the gut, as well as extensive leaky gut. So this is really confirming that likely the pathogens that we're detecting on that first page are probably having a big impact. The other thing to note about pathogens, such as E. coli pathogens and Salmonella, is that they can not only cause inflammation, but they also thrive in an inflammatory environment. So that's kind of one of those additional sort of vicious cycles. So again, kind of connecting all the dots here, we can certainly see that pathogens seem to be causing not only symptoms, they're consistent with the symptoms the patient is having, but also that we see direct evidence on the test of a likely impact from these pathogens. But we also see that there are some predisposing conditions that make it more likely that these pathogens can thrive. So that's really kind of this new perspective on how we can use this information for some additional therapeutic support. So again, this is just recapping the case. Of course, this is not Pseudomonas, it was Salmonella and E. coli. It was a 22-year-old male, generally fit and health conscious, complained of kind of mild diarrhea off and on, as well as mild abdominal pain. So nothing really particularly acute, nothing particularly intense like we would expect from typical food poisoning, plus recent international travel. So the case summary, there was Salmonella, enterohemorrhagic E. coli, both high. H. pylori at a moderate level, plus an overgrowth pattern suggesting digestive dysfunction. Particularly important to be the low Faecalibacterium prausnitzii and Bacteroidetes, because we know they can produce factors that help counteract these pathogens. We also see high calprotectin, high zonulin, and occult blood, providing significant evidence that these pathogens are having a negative impact. So this really kind of comes back to one of these key concepts we talked about last time, which is when we're looking at inflammatory scenarios with inflammatory microbes, and especially when we have confirmed inflammation with, in this case, high calprotectin, as well as barrier disruption from the high zonulin, there's really a kind of a balance with the beneficial microbes. And that can kind of, again, be this two-way interaction where the beneficial microbes are having an anti-inflammatory effect, plus they also help to protect the intestinal barrier. And they also have to directly counteract some of these pathogens. So we're looking for that balance. And typically when there's not enough beneficial microbes, that makes it more likely that the inflammatory microbes can really gain a foothold. So the idea is you want to just kind of shift that balance back in the other direction. So the supportive approach typically in these types of scenarios is to consider antimicrobial botanicals. Of course, whenever there's serious infections, that may also be a scenario where further evaluation by a specialist is required. But also there's lots of evidence that various natural approaches can be supportive, not only in counteracting these types of pathogens, but also promoting a healthier environment. But antimicrobial botanicals can certainly be helpful. Taking an anti-inflammatory type approach in terms of diet and supplement approaches can be very helpful, as well as leaky gut support. In particular, really supporting those beneficial bacteria. When it comes to the butyrate producers, that largely comes down to fiber in the diet and supplementation, and also considering things like polyphenols that can support their growth, as well as butyrate supplementation, and then various dietary modifications. So we talked earlier in the example of C. diff that there's now growing evidence that excessive amino acids or proteins in the colon can actually shift dysbiosis in the direction towards inflammatory bacteria. So this is another area you may want to look at. Is this person consuming high amounts of protein? Are they not digesting protein well? In this case, we did see H. pylori was high, or at least was moderate, suggesting that this may have an impact on protein digestion. So again, this is really kind of, when we're looking at how do we interpret GI map and how do we get the most out of it, it's very helpful to really understand these basic dysbiosis patterns and then how they interact in terms of how do the pathogens and opportunists sort of play into that? How do they cause these dysbiosis patterns? And then also how do these altered physiological imbalances play into this as well? So on the next slide, again, this is kind of coming back to our key concept that we were focusing on in this webinar, that there can be this extensive interaction between the various dysbiotic pathogenic microbes and then altered gastrointestinal physiology. And understanding those details can really go a long way towards helping you target the type of approach that you want to take for a given patient or client. So once again, it really comes back to learning how to connect the dots with GI map so you can get the most out of it. So that brings me to the end of the webinar, and I would encourage you to check out some of our learning resources at diagnosticsolutionslab.com. We have extensive supportive information, interpretive guides, and particularly videos from recorded webinars, etc., that cover many of these topics and help you understand how to get the most out of GI map. So with that, I thank you for listening today, and welcome any questions you may have.
Thank you, Tom. I have been trying to write down the questions on the side as they came along. One was in regards to Akkermansia, and so on the one of the examples you had, there was a below detected levels, but that's not marked as red. Is there a reason for that?
Yes, yeah, that's unfortunately just kind of a fluke of the software that's used to generate the reports. The software needs a number to flag it as low. If it just says less than DL, it's not able to recognize that. So we're hoping to get to have a new solution at some point. So we, in our education materials, we of course emphasize that that is very low and it's below detection limit. And so that's just something that clinicians need to be aware of, particularly in that normal bacteria flora section, because most of those bacteria should be within their normal range and not too low. But yes, that's something we need to get that corrected at some point, because some clinicians do miss that until they've been educated on that more.
That opened up for another question, and I think it was an Icelandic practitioner, and I see it a little bit in Denmark as well, that the patients do have high Akkermansia. And Absolutely. Then of course, that's, what do we do with that kind of, because we want the patients to have Akkermansia, but can you comment on that, please?
It's kind of the typical scenario with most of the beneficial bacteria, where you don't necessarily want them too high or too low. Now with Akkermansia, there's not a lot of direct evidence that high levels cause too many issues. There is some concern because it's a mucus degrading bacterium, that if that's too excessive, if you have very high levels of Akkermansia, they may be degrading too much mucus that can eventually lead to a depletion of the mucus. Clinically, we only see that kind of at the very higher end of the ranges. So when we see it up, say, above maybe E7, E8, when it's in the more E5, E6 range, it's more consistent with just digestive dysfunction. Now, we don't really know exactly why Akkermansia is overgrown in that scenario. I can kind of speculate a little bit, but there's really no research indicating why Akkermansia may be overgrown with digestive dysfunction. So for now, it's just something we recognize is usually being there with that digestive dysfunction pattern. And maybe due to the fact that when you don't digest well, more carbohydrates get into the colon and increase fermentation. So you might get, in some scenarios, more short-chain fatty acids like butyrate temporarily. And then we know that short-chain fatty acids stimulate mucus production. So maybe you get a higher mucus production that just supports more Akkermansia when you're not digesting well. But there's really not good research supporting that.
Okay. Another question came up in regards to the Epstein-Barr markers that you have, if they are lytic or dormant phases when you are detecting them. Do you know that?
What we typically see, so certainly it's not a diagnostic test in any way for those two viruses, CMV or EBV, because they usually are, for example, dormant in immune cells, et cetera, internally. So you can have EBV and CMV and it just doesn't show up in the stool. Now what happens, it's a little bit different in each case, but with EBV, in some patients, it can actually infect the intestinal epithelium. That's not that common, but in some patients it can. So presumably when the intestinal epithelium is shed, and that's, turns over every 3 to 5 days or so, then we will detect it if it's active and growing. And we do have some evidence from research suggesting that EBV that is in the intestinal epithelium, presumably when we detect it, that can have some negative impacts on the intestinal barrier. So it's kind of further support that there might be some issues with the intestinal lining, intestinal health, when EBV is detected in the stool. And then for CMV, it's a little bit different. It can be reactivated in monocytes, which is a type of immune cell. Those can travel then, once it's reactivated, the CMV is reactivated, they can travel to the gut and then become inflammatory macrophages. So mostly clinically, it's been linked to potentially worse scenarios and worse inflammation for ulcerative colitis patients. And I have seen that on a couple occasions with certain cases. Generally, though, it's thought to be indicative that CMV was reactivated and that it's targeting the gut, and that can lead to increased inflammation in the gut when we detect it. We don't see either of those very often, though.
Super. So then there's been quite a few questions in regards to H. pylori, and it could be a whole webinar by itself, but it's questions to like, does low stomach acid or high stomach acid increase the risk of H. pylori? And should you give like stomach acid betaine HCl when treating H. pylori? I think questions like that. So could you comment on that, please?
Yes, it's a little bit of a complex scenario when you get into the details. The majority of people that have chronic H. pylori tend to have low stomach acid. Now, in a subset of cases with H. pylori infection, people can have elevated stomach acid. It depends on specifically where in the stomach it's infecting. So that's one of the factors, but that's a minority of cases, and we don't see evidence of that, at least from what we can tell, very frequently. As far as susceptibility based on stomach acid being altered, it's not entirely clear. We do know that C. diff itself, I mean, one of the reasons why it suppresses stomach acid is because it's trying to create a better environment for itself. So presumably if you already have low stomach acid, then that may create a more favorable environment for overgrowth. Mostly it comes down to, though, the ecosystem in the stomach. So if you do have low or high stomach acid, that could affect the ecosystem. And anytime you disrupt the ecosystem, such as lactobacillus, which is kind of the normal healthy dominant microbe, along with streptococcus in the stomach, once you disrupt that, then it can overgrow. And there's many scenarios that could potentially disrupt that. So to my knowledge, there's not a lot of research addressing acid sort of high or low being a scenario that increases susceptibility. From what I understand, it's mostly through affecting the ecosystem. Now, as far as supplementing with HCl when H. pylori is present, most clinicians, as a rule, will avoid it because a precursor to the low stomach acid is thought to be the gastritis. So the low-grade inflammation in the stomach is what often is thought to lead to the hypochlorhydria. So if you have inflammation in the stomach, that's thought to erode the protective mucus layer. So essentially the stomach lining is not as healthy when it's inflamed, so it's not as able to protect itself against the effects of acid. Clinically, some clinicians have mentioned that they do still kind of on a trial and error basis use HCl and probably titrate it. And they base sort of that on symptoms. So obviously if symptoms, patient gets negative symptoms, then they will stop supplementing. But the majority of clinicians that I've talked to tend to not use HCl when H. pylori is present.
That would be like putting acid into the wound or something.
Yeah, certainly when there's significant, obviously if H. pylori is causing ulcers, then that's a pretty obvious scenario. But even the more subtle gastritis, there may still be a problem with that. And some clinicians have noted that can exacerbate symptoms.
Super. Thank you for that explanation. And it just shows how it's not that simple always and how we have to try ourselves a little bit around with the patients and see how they respond.
It'd be easier to answer questions if things were simple.
Yeah.
I try to give the simplest answer I can, but also not be misleading at all.
So I think maybe the last question is in regards to yeast. Because now with bacteria, you're talking about SIBO, and there's a small intestine and the large intestine and so on. And then there's yeast. And then there's this thing about yeast deteriorating in the tube lab. Maybe you can't detect the yeast, but some labs use a microscopy. Can you hear me?
Actually, you had just cut out there for a moment.
Yeah,
Do you mind
repeating? I'll just repeat. Yeah, I'll repeat it. So that there's this question about the yeasts like degrading in the tubes on the way to the lab, and then like historically a lab would do a culture on yeast and then they would look in microscopy, but because it's only or not only, but PCR, it's a different way. So are there any concerns in regards to a possible negative or false negative yeast infection or overgrowth?
That's a good question. So generally we find the opposite with PCR, that on other tests, so I've seen some direct comparisons on a split sample with other labs that just do microscopy. And at least in some of those cases that I've seen, they were negative for yeast, including Candida, based on microscopy, but positive on PCR. So that's probably the more common scenario. Theoretically, yes, there can always be a chance for false negatives. There may be some factors like that you mentioned. I'm not one of the lab technicians, so I don't know the details of sort of how all these things play out in the lab, but generally, if yeast is present, it can be present anywhere in the GI tract, especially in the oral cavity. So you can have yeast that's not necessarily detected either by microscopy or PCR. If there's a certain amount in the stool, it's highly likely to be picked up. So that's really the key, is there has to be a certain amount in the stool. And the sensitivity PCR is very, very good. I don't know specifically what it is for our markers for Candida. But a good example would be for H. pylori. I know we can detect down to 10 cells per gram of stool. And those results are fairly consistent across microbes. They vary a bit. So that's really one of the variables, is just where the overgrowth is. So you'll see that a lot of clinicians say, well, we've done an organic acids test and the markers on that test were positive, but on stool tests it's negative, whether it's PCR or microscopy. And that could be because Candida is overgrown in a place that's higher up, such as the oral cavity, stomach. It can exist even in the stomach. So there's a lot of details there, but so the short answer is yes, there can be a potential for a false positive, but if it's in the stool above a certain small amount, then PCR is highly likely to detect it.
Thank you very much, Tom. It has been a pleasure. As usual, you are an amazing speaker, and we could carry on and many questions comes up, and I've tried to get them together here and so on. But please, everybody, the recording will be shared as soon as possible. Remember, next week, again, same time, same place, on antibiotic resistance, and we'll find, just go more into details in the invitation that I quickly have to make tomorrow. So Tom, thank you very much for your time and your personality, and thanks for sharing, and I'll give you a virtual hug and everything that goes with it.
Okay, well, thank you so much. It was a pleasure, and I hope everyone has a great day.
Take care.